Hello everyone, today we will discuss about polymerase chain reaction and also known as PCR. Now what is it? It is a biochemical technology in molecular biology to amplify a single or a few copies of DNA sequence to millions of copies of that particular DNA sequence. So this idea was given by Mullis, and for which he was awarded Nobel Prize in 1993. We should understand how the term polymerase chain reaction came. The polymerase enzyme, DNA polymerase enzyme, is used in DNA replication. So, to multiply the DNA in PCR also, we use polymerase enzyme as it is a chain reaction, the term polymerase chain reaction was given to it. Now, how this exponential amplification of the gene in PCR takes place is, you can see over here that there is a template DNA and there is a pink portion, red portion of this, which is a wanted gene which we want to study. So in the first cycle, we can see multiple copies are being prepared in various cycles, and how we will calculate that how much copies will be formed after how many cycles is just by a simple formula, that is 2 raised to the power of n, where n is the number of cycles. So like in the fourth cycle we can calculate 2 over 4, that is 32 copies. This is how exponential amplification of the gene takes place in PCR, and we can get the amount of DNA we require to study. Now, how will the basic procedure will take place of PCR? We will discuss this procedure in detail also. Just briefing, what is the PCR? Firstly, we will locate the target DNA sequence which we want to study. In this, the purple and the blue part, this is the target, the DNA in between this part is the target DNA sequence. Okay, if we have determined which DNA sequence we want to study, after that the step 1 comes is denaturing. The denaturation is with the help of heat. Okay, in this the strands are separated and single stranded DNA is formed. Second step is annealing. In this, what we have added is, we can see we have added two things. One is purple thing and one is blue thing. That two things, these are known as primers. They will start the replication from the part we want it to start. It will bind to that sequence where we want it to bind. Then the step 3 is, we will add an enzyme known as Taq polymerase. This is DNA polymerase, but this one is heat resistant. Okay, and then the replication will take place by adding of all these things, and along with we will add nucleotides. Then extension and elongation will take place, and we will receive our desired DNA segment. Okay, again repeating it, we will discuss it in very detail also. If this is the original DNA to be replicated, firstly denaturation must be done. We will separate them. Then annealing must be done, in which the primers, this is the primer which is added. Okay, this primer must have complementary DNA nucleotides to the segment where it has to attach. Then elongation is there, and then these cycles are repeated as many times we want. Okay, now for the basic PCR, what things we will require? They are DNA template. Firstly, we require the region. We have to locate the region which we need to be amplified. Then we require 2 primers. These primers should be complementary to the 3' ends of each coding as well as anti-coding strand of the DNA target, so that the specificity of the DNA segment is maintained. Then we require an enzyme known as Taq polymerase. Specifically, it is received from a bacteria uh, because we need a heat stable DNA polymerase. We want it to remain active despite we will repeatedly heat it during many cycles of amplification. And also we will require the building blocks. How elongation will take place is because of the nucleotides. We have to add nucleotides. We will add 4 nucleotides. Okay. And then we will require magnesium chloride, which is a co-activator of Taq polymerase and also buffer solution to maintain that environment. Okay. Now we will discuss in detail the procedure of PCR. So first, as we discussed, is the denaturation step. In this, the heating takes place. Now this all takes place in an instrument known as thermal cycler, in which the tubes are inserted. Okay. In denaturation step, there is heating reaction to the temperature of 94 to 98 degrees Celsius for 30 seconds. What will it do? It will disrupt the bonds between the complementary bases and it will lead to single-stranded DNA. The DNA which was double-stranded will convert itself into single-stranded. Second is the annealing step. In annealing step, what will take place? The temperature is now lowered down. We will lower it around 55 degrees Celsius for 20 to 40 seconds, and then we will add the primers which have been already added to it will now anneal to the single-stranded DNA template. Okay. Third step is the extension or elongation step. In this, DNA polymerase enzyme, the Taq polymerase enzyme, has an optimum temperature to work. It is around 72 degrees Celsius, around 75 to 80 is also good. To remember, it is 72 degrees Celsius and it will work at this temperature. So we will bring the temperature, we will raise again the temperature to 72 degrees Celsius, and now what will it do? It will synthesize a new DNA strand with the help of we are adding the nucleotides. It will synthesize a new DNA template strand, and it will be always the addition will be in the direction of 5' to 3'. Okay. Now to again summarize this is a patient's parent DNA, and this we want to study. Okay, first the step of denaturation is done. We can see here it is double stranded, it is helical double stranded. Now we will separate it. The 2 DNA strands have been received over here. Then the primers, the DNA polymerase and the nucleotides, these are entered, and we will go to second step, which is known as annealing, which is working at 55 degrees Celsius. The temperature must be remembered. It is exactly the same temperature which should be used. Otherwise, the thing will not work. The step 2 annealing is done. In this we can see the primers are added. These are the primers, and they will limit that the segment from here to here is only replicated in various cycles. Then step 3 is the last step, is the elongation. Okay, and it works at 72 degrees Celsius. Now the enzyme Taq polymerase will start working and it will cause the replication from 5' to 3' end. Okay, we can see the replication is in this direction and is in this direction. This is 5' to 3' end. And now we can repeat this cycle many times to the time we want it to repeat. We can calculate. I have told you already 2 raised to power n. When we will think that we have received our amount of DNA which we are required for studying, then we will stop this cycle. Okay, this was the basic of PCR. I will discuss in the later videos the types of PCR. Do like, share and subscribe to my channel. Thanks for watching it.